Can I start?
Yes, please go ahead. You can start. The meeting is being recorded.
Good evening, senior colleagues and colleagues. I'm Olawaju AB, and I'm representing embryonic development of the heart. Following this outline. So by way of introduction, heart is the first functional organ to form and begins to pump blood early in embryonic life, ensuring the distribution of oxygen and nutrients vital for growth and survival. The process of heart formation involves complex cellular, molecular, and genetic interactions. Disruptions at any stage can lead to congenital heart defects, which are among the most common birth defects worldwide. So using the formation of the primary heart field. The vascular system appears in the middle of the third week when the embryo is no longer able to satisfy its nutritional requirements by diffusion alone. Because prior to third week, the embryo depends on the mother's nutrients. But as the embryo develops, the mother's nutrients is unable to sustain its nutritional requirement by ordinary diffusion. Thereby, vascular system appear in the middle of third week to help in this particular function. Progenitor heart cells lies in the epiblast, adjacent to the cranial end of the primitive stick. It later migrates through the streak and into the splanchnic layer of the lateral plate mesoderm, where they form a horseshoe of cluster cells called the primary heart field, cranial to the neural folds. Progenitor heart cells migrate and form the primary heart field on days 16 to 18, and they are specified on both sides from lateral to medial to become the atrial, left ventricle, and most of the right ventricle. Summary here is that from the primary heart field, which is formed on the 16th and 18th day, it is this primary heart field that give rise to both atria, the left ventricle, and most of the right ventricle, meaning that there is part of the right ventricle that is not formed from the primary heart field. So the remainder of the heart, which include the part of the right ventricle and outflow tracts, involving the conus cordis and truncus arteriosus, are derived from the secondary heart field, which is formed on the 20th to 21st. And the secondary heart field resides in the splanchnic mesoderm, which is ventral to the posterior pharynx, and responsible for the lengthening of the outflow tract. Cells in the secondary heart field exhibit laterality, such that those on the right side contribute to the left of the outflow tract region, and those on the right contribute to the left side of the outflow region. And this is what is meant by laterality. So the laterality causes spirally nature of the pulmonary artery and aorta, which ensures that the aorta exits from the left ventricle and pulmonary artery exits from the right ventricle. And this is the significance of laterality. This is a figure showing what has been said in the previous slide. As looking at this, where we have the primitive strip, and the progenitor heart cells migrate to this particular area, which is being referred to as the primary heart field. And this is where the both atrial and left ventricle and parts of the right ventricle originated from. This is further explanation telling us at the lateral part of the mesoderm where we have the primary heart field. So this is another diagram showing what the secondary heart fields look like and the relationship between the secondary heart field and the pharyngeal arches and also the outflow tracts. By looking at this particular diagram, it shows that the secondary heart fields lies in the splanchnic mesoderm at the posterior of the pharynx. So this is the pharynx and this is the secondary heart field. Looking at the relationship, we realize that the secondary heart field is located posterior to the pharynx. And apart from that, it is also being discovered that the secondary outfield provides cells that lengthens the outflow tract region. This is the outflow tract region. So it is the cells in the secondary outfields that help to lengthen this particular outflow region, of which, if there's defects in this lengthening, there's particular outcome that is being seen, which vis-a-vis, meaning that those things that have been formed from the secondary outfield will not definitely come to play. And the fibroblast growth factor has been said to be implicated in this particular function. Primary outfield are induced by the underlying pharyngeal endoderm to form cardiac myoblasts and blood islands, which form blood cells and vessels by the process of vasculogenesis. The islands unite and form a horseshoe-shaped endothelial line tube surrounded by myoblasts known as cardiogenic region. The intraembryonic cavity later develops into the pericardial cavity. Other blood islands appear bilaterally, parallel and close to the midline of the embryonic shield. These islands form a pair of longitudinal vessels that is being called dorsal aortae. Now, considering the laterality and heart defects, establishing laterality during gastrulation is essential for patterning the right and the left side of the heart. Distortion of laterality pathway can result in dextrocardia, VSD, ASD, double outlet right ventricle. It has also been said that it can lead to outflow tract effects, such as defects, such as transposition of great vessels and pulmonary stenosis. There can also be atrial and ventricular isomerism. Likewise, inversion can also occur if laterality has not taken place. It is of note that serotonin is part of the key molecule, helps in initiating this particular pathway. And intake of selective serotonin inhibitor have been linked to heart defects, whereby disrupting signaling in the laterality pathway. Formation and position of heart tubes. Initially, the central portion of cardiogenic area is anterior to the oropharyngeal membrane and the neural plates. With growth of the brain and cephalic folding of the embryo, the oropharyngeal membrane is pulled forward, while the heart and the pericardial cavity moves first to the cervical region, then finally to the thorax. As the embryo grows, it bends cephalocaudally. It also folds laterally. The caudal region of the paired cardiac tube merges, except the caudal-most area. The central part of the horseshoe-shaped tube, expanded tube consisting of an inner endothelial lining and outer myocardial layer. The caudal pole receives venous drainage and begins to pump blood out of the first aortic arch into the dorsal aorta at its cranial pole. The development of heart tube budges more and more into the pericardial cavity. And initially, the tube remains attached to the dorsal side of the pericardial cavity by a fold of mesodermal tissue, which is known as the dorsal mesocardium. It should be noted that no ventral mesocardium is ever formed. It is the dorsal mesocardium that is being formed. With further development, the dorsal mesocardium disappears, creating transverse pericardial sinus. This is the one that connects both sides of pericardial cavity. The heart is now suspended in the cavity by blood vessel at its cranial and caudal poles. And it is during this event, the myocardium thickens and secretes a thick layer of extracellular matrix, rich in hyaluronic acid, that separates it from the endothelium. So in addition to this, mesothelial cells on the surface of the septum transversum form the proepicardium near the sinus venous at its caudal end and migrate over the heart to form most of the epicardium. and the remainder of the epicardium is formed from mesothelial cells originating in the outflow tract region. And the air tube consists of 3 layers. First is the endocardium, forming the inner endothelial lining of the heart, followed by the myocardium, which forms the muscular wall, and the last one, which is the epicardium or the visceral pericardium. This covers the outside of the tube. And it is also this outside layer that is responsible for the formation of coronary arteries, including the endothelial linings and the smooth muscle. This particular figure is only emphasizing the fact that the blood island or the cardiogenic area is anterior to the oropharyngeal membrane initially before the changes occur as has been described in the previous slides. Now, formation of the cardiac loop, which is also an important step in the development of the heart. There's elongation of the heart tube at the cranial end from the secondary heart field, leading to normal formation of the right ventricle and the outflow tract region. Cornus cordis and truncus arteriosus that form parts of the aorta and the pulmonary artery, and for the looping process. As the outflow tract lengthens, the cardiac tube begins to bend on day 23. The cephalic portion of the tube bends ventrally, caudally, and to the right, while the atrial portion, which is the cauda, shifts dorsocranially and to the left. This bending, which may be due to cell shape changes, creates the cardiac loop, which is completed by day 28. And while the cardiac loop is forming, local expansion becomes visible throughout the length of the tube. The summary of this particular one is that there is lengthening of the cardiac tube, and this particular lengthening starts with the outflow tract lengthening at day 23, and the cephalic part later now bends anteriorly, then caudally to the right. These are the 3 things that combines the first part of the cephalic portion, while the caudal portion bends posteriorly and cranially then to the left, because at times this can be mixed up. We should just know the way it's matched. When there's difference in lengthening, this will cause double outlet right ventricle. It can also cause ventricular septal defects, tetralogy of Fallot, pulmonary atresia, and pulmonary stenosis. The atrial portion, which initially is a paired structure outside the pericardial cavity, forms a common atrium and is incorporated into the pericardial cavity. The atrioventricular junction remains narrow and forms the atrioventricular canal, which connects the common atrium and the early embryonic ventricle. The bulbus cordis is narrow except for its proximal third. This portion will form the trabeculated part of the right ventricle. For the mid portion, the cornus cordis, this forms the outflow tracts of both ventricles. The distal part of the bulbus, known as the truncus arteriosus, this forms the roots and proximal portion of the aorta and the pulmonary artery. The junction between the ventricle and the bulbus cordis externally indicates by the bulboventricular sulcus remains narrow, and this is called the primary intraventricular foramen. So at the end of the loop formation, the smooth wall heart tube from primitive trabeculae in 2 sharply defined areas, proximal and distal to the primary interventricular foramen. So the bulbus temporarily remains smooth-walled. The trabeculated primitive ventricle is called the primitive left ventricle. Likewise, the trabeculated proximal third of the bulbus cordis is known as the primitive right ventricle. The conotruncal portion of the heart tube initially on the right side of the precardial cavity, shifts gradually to a more medial position. And this change in position is a result of formation of two transverse dilation of the atrium, bulging on each side of the bulbous cordis. So this is a diagram that is showing the looping process and the division of the tube, the arch tube. Looking at it, we have different portions. We have the atrial part, or this particular part that the cursor is, which is known as the sinus venosus. And followed by it is the atrium. It's still the same color, however, it is the proximal part that is being referred to as the sinus venosus. So this is followed by the ventricle, and we can also see the bulbous cordis. Truncus arteriosus is at the tip, which is still the one that is being followed by the bulbous cordis. So initially, the arch tube is usually a long, straight tube, but as looping occurs, we realize that it later becomes to bend to form the C pattern as it is being described in the previous slide, talking about the caudal part as it moves ventrally and to the right, while the other part moves cranially and posteriorly to the left. And this finally assumes the S shape. And this particular diagram is also showing the bulbous cordis as it's being divided. Looking at this diagram, we see that this is the bulbous cordis, and the broken line is representing the pericardium, while this part is the left ventricle and this is the primitive left atrium. But what this particular diagram is showing is telling us about the division of the bulbous cordis, which is divided into three. We have the truncus arteriosus, we have the conus cordia, and the half part, which is the trabeculated part of the right ventricle. These are what the bulbous cordis later divide to. So formation of the cardiac septa. The major septa of the heart are formed between the 27th and 37th day of development, and when the embryo grows in length from 5 millimeters to approximately 16 to 17 millimeters. A septum is formed involving two actively growing masses of tissue approaching each other until fusions occur, dividing the lumen into two separate canals. Formation of tissue masses depend on synthesis and deposition of extracellular matrices and cell proliferation known as endocardial quotient that develop at the atrioventricular and conotruncal region. So this particular diagram is also showing us how the septum is being formed by growth of the opposite ridges. These are the ridges, and it's coming together. Looking at B and at C is almost opposite the other end of the ridge. Abnormalities in endocardial quotient formation can cause cardiac malformation, such as atrial and ventricular septal defects, and also cause defects involving the great vessels, such as transposition of the great vessels, common truncus arteriosus, and tetralogy of Fallot. For septum formation of common atrium, at the end of the 4th week, a sickle-shaped crest grows from the roof of the common atrium into the lumen. And this crest is the first portion of the septum primum. The two limbs of this septum extend towards the endocardial quotient in the atrioventricular canal. And the opening between the lower rib of the septum primum and the endocardial quotient is known as the ostium primum. With further developments, extension of the superior and inferior endocardial quotients grow along the edge of the septum primum, closing the ostium primum. Before this closure is completed, Cell death produces perforation in the upper portion of the septum primum. The coalescence of these perforations forms the ostium secundum, thereby ensuring free blood flow from the right to the left primitive atrium. So this is a diagram that is showing what is being said in the previous slide. As we have the septum primum, and looking at it here, as we have the septum primum, this is the left atrium, right? This is the right atrium. This is the right ventricle. This is the right atrium too, and we also have the left ventricle. Again, this is the right atrium. We have the left atrium, and the communication, the presence of atrium primum allows the blood flow between the right atrium and the left atrium. The endocardial cushion is here for the right, while this is the left endocardial cushion. In between here, we have the atrioventricular canal. The other diagrams is also self-explanatory, and we can see the labeling as being shown. Abnormalities of atrial septums can lead to core triloculae biventricularly, which means there is complete absence of atrial septum. And again, we can have patent foramen ovale, which is the premature closure of the foramen ovale, leading to massive hypertrophy of the right atrium and the ventricle, and under the mesenchymal cushions. The atrioventricular endocardial cushions appears at the anterior and posterior border of the atrioventricular canal. And initially, the atrioventricular canal gives access only to the primitive left ventricle and is separated from the bulbus cordis by the bulboconoventricular flank. And by the end or near the end of the 5th week, posterior extremity of the flank terminates almost midway along the base of the superior endocardial cushion and is much less prominent than before. So since the atrioventricular canal enlarges to the right, blood passing through the atrioventricular orifice now has direct access to the primitive left as well as the primitive right ventricle. And in addition to the anterior and posterior endocardial cushions, the two lateral atrioventricular cushions appear on the right and the left border of the canal, as been shown in the previous slide. The anterior and the posterior cushions, in the meantime, project further into the lumen and fuse, resulting in a complete division of the canal into the right and the left atrioventricular orifices, and at the end of the 5th week. So this particular figure is showing the formation of the septum in the atrioventricular canal and showing different days of development. Looking at this is a common atrioventricular canal, and at this particular part, as it is growing, there is superior endocardial cushion here, and we can see this indentation trying to divide it. And this progresses until it is fully divided into the right atrioventricular canal and the left atrioventricular canal. If this particular process is not it doesn't take place, what will happen? There will be persistent atrioventricular canal, and as a result, it's from the failure of fusion of the atrioventricular cushion with a defect in the cardiac septum, meaning that we won't have this division, it will just be left with this common atrioventricular canal. Now, atrioventricular valves. After atrioventricular endocardial cushions are fused, each atrioventricular orifice is surrounded by local proliferations of mesenchymal tissue. When the bloodstream hollows out and thins tissue on the ventricular surface of the these proliferations, valves form muscular cords and finally in the course degenerate and is replaced by dense connective tissue. The valves then consist of connective tissue covered by endocardium. They are connected to thick trabeculae in the wall of the ventricle, the papillary muscles by means of chordae tendineae. In this manner, two valve leaflets consisting of bicuspid valve, which form in the left atrioventricular canal, and the three constituting the tricuspid valve that is formed on the right side of the atrioventricular canal. This is showing the formation of atrioventricular valves and chordae tendineae. You can see the perforations that has occurred in the myocardium. And it is from this that the particular papillary muscles, chordae tendineae, and the atrioventricular valves appears. So the valves are hollowed out from the ventricular side, but remain attached to the ventricular wall by the chordae tendineae. When there is defect of the atrioventricular valve, we can have tricuspid atresia, can also have, which is characterized by obliteration of the AV orifice, characterized by absence of fusion of the tricuspid valve, can also be associated with patency of oval foramen, ventricular septal defects, underdevelopment of the right ventricle, and hypertrophy of the left ventricle. Defects of this particular valve can also result in Epstein abnormality, whereby tricuspid valve is displaced towards the apex of the right ventricle. And as a result of that, the valve leaflets are abnormally positioned with the anterior usually enlarged and is being characterized by hypertrophy of the right atrium with a small right ventricle. For the semilunar valves, when partitioning of the truncus is almost complete, primordia of the semilunar valves becomes visible as small tubercles found on the main truncus swellings. One of each pair is assigned to the pulmonary and aortic channels respectively. A third tubercle appears on both channels opposite to the fused truncus swellings, and gradually the tubercles hollows out at upper surface, forming the semilunar valves. And recent evidence has shown that neural crest cells contribute to the formations of these valves. So this is the longitudinal section through the semilunar valves at various weeks. The first one is at the 6th week, second is at the 7th week, while the last one is at the 9th week. And we can see how the semilunar valves develop. From A, you can see it's more hollow there before we get to the final part, which is a C. When there is defect of the semilunar valve, we have the pulmonary, can lead to pulmonary valve stenosis, which is either narrowing or atresia of the valve. And also be aortic valve stenosis, fusion of the thickened valve may be complete with a pinhole appearance. Can also have aortic valve atresia, which is also from the obliteration of the valve. The aorta, left ventricle, and the left atrium may also be markedly underdeveloped. And we may also have the ectopia cordis. Looking at the septum formation in the ventricle, by the end of the 4th week, the two primitive ventricles begin to expand. This is accomplished by continuous growth of the myocardium on the outside and continuous diverticulation and trabecular formation on the inside. The medial walls of the expanding ventricles become opposed and gradually merge, forming the muscular interventricular septum. Sometimes the two walls do not merge completely, and a more or less deep apical cleft between the two ventricles appears. The space between the free rim of the muscular ventricular septum and the fused endocardial cushions permits communication between the two ventricles. So the interventricular foramen above the muscular portion of the interventricular septum shrinks on completion of the conus septum. And during further development, outgrowth of tissue from the anterior, which is the inferior endocardial cushion along the top of the muscular interventricular septum, closes the foramen. And this tissue fuses with the aborting part of the conus septum. Complete closure of the interventricular foramen now forms the membranous part of the interventricular septum. Showing that we have two parts of the interventricular septum. We have the muscular part and we also have the membranous part. And the membranous part is formed following the complete closure of the interventricular foramen. When there is defects of ventricular septum, this will obviously lead to VSD and can also lead to ventricular tetralogy of Fallot, because VSD is also a component of this particular defect, which is the tetralogy of Fallot. Yeah, this particular diagram, the first part is showing the normal heart, while the second one, which is the B, is showing the isolated defect in the membranous part of the interventricular septum. When we look at it, this place is closed, but looking at this part, there is a defect here, and blood from the ventricle flows from the right through the interventricular foramen, as been indicated in this particular diagram. This is also another diagram that is showing the presence of tetralogy of Fallot in this particular diagram. Looking at it, we can see that the four components of tetralogy of Fallot is present here. We have the overriding aorta, we have the pulmonary stenosis, we also have the VSD, which is pronounced here, and the right ventricular hypertrophy. Now, moving to formation of the conductive system of the heart. Initially, the pacemaker of the heart lies in the caudal part of the left cardiac tube. Later, the sinus venosus assumes this function, and the sinus is incorporated into the right atrium. Pacemaker tissue lies near the opening of the superior vena cava, and thus the sinoatrial node is being formed. The atrioventricular node, the AV node, and the bundle of it are derived from two sources. One is the cells in the left wall of the sinus venosus, and two is the cells from the atrioventricular canal. And once the sinus venosus is incorporated into the right atrium, these cells lie in their final position at the base of the interatrial septum. So, summary is that SA node is being formed after the SA node is being formed, following after the sinus has been incorporated into the right atrium. And for the AV node and the bundle of the heart is one of the first organs in developing embryo to form and function. By the start of 4th week, a primitive heart has begun to pump blood, and by the 7th week, development of the heart is complete. The heart developed from the middle embryonic tissue layer known as the mesoderm, and it begins as a tube that joins with blood vessels also forming in the mesoderm. Other mesodermal cells form a coat around the heart tube and become the muscular wall, or which is otherwise known as the myocardium. Thank you. Hello, I hope I'm audible.
Thank you very much, Dr. Abdulaziz. Thank you for doing your presentation. At this point, we'll elicit questions from participants and colleagues, but whilst we wait for that, seniors on the call, consultants, professors on the call, if you have any contributions to make, let's take the contributions, then we can also take questions from our colleagues on the call. Thank you. Hello?
Yeah, you're audible.
Please, any questions, contributions concerning this presentation, please?
Hello, thank you, Dr. Olaiwaju. I don't know, it seems my network had some challenges and I was knocked off towards the end. But I believe you're done, right?
Yes, sir.
Okay. Thank you very much for a very excellent and lucid presentation. Thank you so much. Please, the class, let's appreciate him. Thank you for kickstarting our curriculum presentation, and it was indeed a wonderful one. We're so grateful. So we'll take questions and comments from the class before we go to our consultants in the house. So I don't know, you can just unmute yourself or raise your electronic hand if you have any question or you want to make a comment. Thank you very much. Okay, I don't know if there's anyone who wants to make a question, who has a question or want to make a comment. Dr. Olaiwaju, let me just ask a question. There's something I didn't really get when you were talking about the fusion of the endocardial cushions. I didn't understand how they really fuse. I don't know whether you can actually go over that explanation. Yes.
You mean this place?
I don't know if yes, is this one exactly? Yes, this one. This
Yes,
one.
this particular figure has helped to explain or show the progression in which the fusions occur. Starting from this particular part, you can see that there is a common intraventricular canal.
Your voice is breaking. I don't know whether it's only me or is.
Am I audible now?
Excellent. You're audible now.
So I said this particular figure has helped to explain or show the progression from the common atrioventricular canal to when it is being divided into the right atrioventricular and the left, where the superior endocardial and the inferior endocardial cushion. Looking at this particular one, you see that it is really round and it is a common atrioventricular canal. But on getting to this particular part, you see this lateral cushion here, and there's indentation of this canal. And we can also see the indentation that is being caused by the superior endocardium, and under the inferior part is also being indented by the inferior endocardial canal. And as this compresses the common atrioventricular canal, you can see that it's now left with a narrowing in between the two parts that want to be divided into the right atrioventricular and the left atrioventricular. And by the time there is complete obliteration of this particular part, you will see that it is now well clear to form both the right and the left as they depicted in this particular diagram. But in all this, we can see that this progression occurs as the development in the embryo occur, because the first one is at day 23. This second one, when we are having a lateral cushion, the superior and the inferior, not yet fully fixed, is at day 26, but it is completely divided into the right and the left atrioventricular canal at day 35 of the embryonic development. Thank you. I don't know, maybe I've been able to answer your question, sir.
Excellent. Thank you very much. I think it's much clearer now. Thank you so much. Okay, I don't know if there's any other person with any question or any comments. Anybody with any question or any comments? Okay, then I'll go to our consultants in the house, Dr. Conte, Dr. Allison, Dr. Kweki, then Prof. I don't know if there are comments. I know that this topic is usually a very important topic as that comes out frequently in the exams.
Hello, good evening. I don't know if I'm
Go
audible.
ahead, sir. Yes, sir.
Yes, good evening. Good evening. We can hear you. Please go ahead.
Hey, Dr.
Yes.
Akin, make some
I don't know if
comments.
it is for me.
Also want to appreciate the venture.
Your voice is
Am I
breaking, sir.
audible?
I don't know if it is my part, sir.
Your voice is breaking, but I think it's more stable now. Go ahead, Chief. Go ahead.
Hello, I can't hear. I don't know if it is my side so that I can.
I'm not actually hearing too. Dr. Ojo, I don't know. We can't hear you, sir. I don't know if you can hear us, or maybe you can type your question on the chat box or your contribution. On the chat box that we can actually follow. Okay, while we wait for Dr. Ojo, I don't know if there are any other questions or comments from our chiefs. Any comments from our chiefs, from Dr. Conte, Dr. Allison?
So Oladimiji's hand is up.
Okay, Prof, sir, please go ahead, sir.
Thank you for this wonderful presentation. Hope I'm audible.
Yes, sir.
Yes, sir, we can hear you, sir.
So one important thing for MCQ sake is to know the pharyngeal arches that give rise to different vascular connections of the heart. You know, those third, fourth pharyngeal arch, what the remnants, what it develops into, is a good MCQ question that usually come out. Number two is for OSCE stations, the clinical correlates of the segments of the embryology. So, for example, they can ask you to quickly describe how intertrial septum forms and discuss the clinical correlates. So you should know all the possible clinical or congenital conditions that can arise from each segment of the formations of the chambers of the heart. For example, now, when talking about, like this slide you have now, talking about the formation of the atrioventricular valve from the endocardial cushion. So what are the possible clinical correlates from endocardial cushion defects? We should be able to state all the clinical conditions that can arise from it. Then you should also be able to discuss the formation of the trunk, which is the pulmonary and aortic trunk. I noticed that you didn't really go in details to describe that or discuss that and the clinical correlate that can arise from it. I'm not sure you really actually talked about formation of the pulmonary and the aorta. Or maybe my slides went off at that time.
No, Prof, he didn't really emphasize on it, but we have someone who's going to discuss the embryology of the great vessels.
Great vessels.
Yeah. Okay, sure. I just want to be sure that the clinical correlates are well known for each chambers, which segments are formed in the heart. So that's the likely questions that can come out in the OSCE station or Vival stations. Thank you very much. So if we may ask you, what are the possible clinical correlates related to interatrial septum formation? Over to you, sir.
Clinical correlates from intrathecal septal formation. So we can have it through ASD. Hello, sir.
Clinical correlate, let me see. Yeah, yes.
Yes,
Can
we can have atrioventricular septum, atrioventricular septal defects from it.
Dr. Lai, clinical correlate to interatrial septal formation.
Maybe I didn't get the question. I didn't understand the question.
If you say atrioventricular septal defects, you only get 1 over 10. So because we'll have expected that you describe every form of atrial septal defects you can have. So you can't do an echo and just write diagnosis.
That's what I can recall for now.
Is that how you write that in diagnosis? Have you?
Yeah.
No, I'll be chief. We've done echoes before. You've seen ESD. I'm sure you've never written diagnosis ESD. You have the different types now, have you?
Okay.
So let's hear you, sir. I don't know if I'm still audible.
Yes, Prof, we can hear you, sir.
This is network.
We can have the, yes, sir. So the types I remember based on what I presented, we have the strong secondum ASD, and we can also have the strong premium ASD. Likewise, I think there's presence of the possibility of having sinus venosum source ASD is also there.
Okay, so Dr. So the examiners would
Yes,
have
sir.
a marking specific, okay? So when they're asking you the possible clinical condition associated with embryology of atrial septal formation, so there are many types, right?
Yes, sir.
So there could be primum, which will be related to endocardial defect, right? So there could be secondum, right?
Yes, sir.
When you mention sinus venosus, there is the SVC, right? Type.
Yeah.
Sinus related to the superior vena cava. There's sinus venosus ASD related to the inferior vena cava, right?
Yes, sir.
So, and there's also what is called coronary sinus atrial septal defect, right?
Yeah.
Hope you understand. And also there's common atrium. That means that there is no even formation of there's failure
The
of the formation
septum
of the
at all.
that is what they will mark. So you just come out and say ASD, you'll just be laughing. You get. So the same, you should also know the converse is also true for ventricular septal defects. So you should know all the types. Because that's what we'll have expected you'll have described as part of your clinical or correlates for each of the segments of the chamber. Okay, sir. Thank you.
Okay, sir. Thank you, sir.
Thank you very much, Prof. We are grateful. We appreciate you, sir. I don't know if there's any more question, any more comments. Thank you once again, Dr. Lariwaju, for an excellent presentation. Please, you will drop the presentation in the WhatsApp group so that we can also read it later. Thank you so much. So I think if the class is okay, we'll go ahead and continue with the MCQs. I don't know whether that's fine. Dr. Eugene, are you here, sir?
On. So I'll stop this. I'll go off then. I'll join again.
All right. Thank you so much.